If you've had a child development course or brain development course, you also know that as children develop, they develop their primary and sensory abilities earlier than, say, higher association abilities. They learn to reach and grab things before they do major problem solving. And brain development goes along with that. This is just looking at the different lobes. Frontal lobe up here, what do we have? Frontal, parietal, temporal, and occipital lobes. And occipital lobe is mostly visual, doesn't have a whole lot of association. You find that, or higher association cortex. And, again, we have the little arrows that point where the peaks are. The peak is actually earlier than this, which here we're looking at 7 to 20-year-olds, roughly speaking. Occipital lobe is already on the decline after that. It's already reached its peak level. Whereas parietal lobe up here is coming in next. Boys have peak here, girls have just peaked already. Parietal lobe is a mix of sensory motor cortex and higher association cortex, but sort of as a ratio, it's not as much association cortex as, say, frontal or temporal lobes. And then the most prolonged development are frontal and temporal lobes, which have the more proportionally large arrangement of association versus sensory or motor cortex. So that goes along with what we know from cognitive development. It also goes along with what we know from post-mortem studies, where we can kind of confirm, well, what is that reflecting? This is just the synapse numbers again. And I've marked out the peaks. They're actually looking at 3 different areas of cortex: visual cortex, auditory cortex, and then a higher level association cortex, prefrontal cortex, which you can see has the latest peak. That synapse number, dendritic branching, will go along roughly with that. And so that may be one of the major contributors to, say, gray matter volumes and the timing of the gray matter volume increases. Okay, well, that's volume. In the case of cortex, we can look in a little more detail. We can look separately at surface area and cortical thickness. If you take just your sort of a plug out of the cortex, you can measure both the radial aspect, or cortical thickness. In a lot of ways, the tangential aspect, the surface area, gives us a lot of our different functional regions. Whereas within a restricted region, you may have, well, you've got things called mini columns for one thing, which may be little processing units on their own. But if we see different rates in the development of surface area, or the expansion of surface area versus changes in thickness, particularly, say, in a patient population versus controls, that might tell us something about what's going on at the cellular level. Differences in thickness, for instance, might suggest an abnormality in neuronal migration. Differences or abnormalities in neuroproliferation could affect thickness, or they could affect surface area. That's partly depending on the timing of any sort of if it's an insult, or the timing of when a relevant gene becomes affected. Might have different effects on those two things. So we'd like to look at both of those. And here we're looking just at birth to 2 years, so that period where we've got very rapid increase in the volume of the brain. This upper and these are looking at rates of change, or rather, sorry, percent of change across that time period. Cortical thickness on the top in 4 different views, and then surface area on the bottom. And what you see for cortical thickness is this works out to be, the green works out to be about a 30% increase in cortical thickness across that time period. And one thing to note is that it's pretty equal across the entire cortex. There's a couple of areas where it's slower in blue. There's a couple areas where it's a little bit faster in yellow and orange. But for the most part, it's fairly consistent around 30%. Surface area, on the other hand, first of all, we're looking at an entirely different scale here. The upper scale is 0 to 60% change, whereas we're looking at up to 150% change for surface area. And you can see a whole lot more spatial differentiation, almost, in those rates. And as we see for volume and for number of synapses and various other measures, we're seeing a slower rate of change, or less change, in, say, primary sensory and motor cortex, yellow also in visual cortex, but the areas that stand out are higher level association. So we've got prefrontal cortex, we've got a bit of parietal cortex, some temporal lobe. So those are still showing quite a bit of expansion in surface area in those first 2 years. And it suggests that a lot of that volumetric increase that we see is due to surface area expansion rather than changes in cortical thickness. One caveat to that is that in MRI measures, we're probably not quite as sensitive to thickness differences, just because cortex varies from something like 3 to 8 millimeters across the cortex. And we're dealing with voxels that are 1 millimeter on a side. So we're not going to be quite as sensitive to that as we can be to surface area. And here we're looking at basically the same thing, but now looking childhood to adolescence, what's going on. 3 to 21 years. And these are showing annualized rates of development. The numbers across the top tell you the age, so 3-year-olds, 5-year-olds, 7-year-olds, on up to 21-year-olds. And then the rows are just different views of the brain. One thing to note is this is a different color scheme than the other one. So here, yellow means the most change, and red is lesser change, whereas the other one was the other way around. But what you're seeing is matching what I showed you in the graphs of total volumes. We see very rapid growth continuing from 3 to 9. Now we're looking at, and this is, surface area, if I didn't say that. But lower rates than we saw in 0 to 2, in birth to 2 years. So it's slowing down. Where'd my cursor go? It's slowing down. And here where you start to see it go almost to black, that's kind of our inflection point. Around, say, 8 to 10 years. And then in blue, we're starting to see a much slower decrease in surface area. And this is actually from the Ping study up at UCSD, and this has about 1,000 subjects in it, so it's a nice large sample, which again you can't do with postmortem. And so that statement that most of our volumetric increase postmortem is from surface area kind of fits also with the postmortem. Keep losing my cursor. So these are the, let me try this. There it is, okay. These are the same images you saw before of dendritic expansion. I don't know if you'll be able to see it at this resolution, probably not. But here we're looking at Nissl stains. So this is looking at the cell bodies. And along with that expansion of the dendritic branching, the dendritic arbors, you're also seeing an increase in the spaces between the neurons over that same age. It's, I think, particularly in the tangential direction rather than the radial direction. So they're starting to spread out a little bit, mostly as you get all of those dendrites in there. But that then helps to explain how you're getting the surface area expansion. But again, that we're getting a bit of an index of that. Okay, so like I said, I was just going to mention a little bit of what we see in autism. Sorry, I got a little pop-up I'm trying to get rid of now. And so, okay, so we're looking at the volume, surface area. That might seem a little bit simplistic in some ways. It's like, okay, well, I've already said there's a whole lot within the voxel. But it actually can be pretty informative with things like developmental disorders. Looking at autism, this is a study from some time ago, here we're just looking at cortical gray volume. This is over the entire cerebrum. from 2 to 16 years of age. And if you look, you can see that in these youngest kids, where open circles are children with autism, there's a substantial increase in the cortical gray matter volume in that age range. In this sample, 2 and 3-year-olds combined as a group had 12% more cortical gray matter than the control group. So something has happened to cause the volume of the cortex to expand, to increase substantially during those first couple of years of life. And that's actually been one of the most consistent findings in brain imaging in autism, is that increase in volume. It's also been found that it's more substantial in higher association areas, so it's more of an increase in frontal and temporal lobes than in parietal and occipital lobes. That's been shown in several studies. This is another one. Red is the autism group from, I think that's up to about 6 years of age. Blue is the control group. And you can see that autism is starting out a bit larger, but then the typical group will tend to catch up. When that's been examined for surface area and thickness, it actually looked like it's a greater expansion of surface area rather than thickness. So that suggests that, so those findings and sort of the replicability of the findings that sort of led to some particular interest in pruning as a possible explanation, or pruning deficits, I should say. One possible explanation for differences for those effects. It could be differences in neuroproliferation. Maybe there's more neurons in this cortex in autism, and you'd want to know why that is and what that might do. And definitely also contributes to the interest in possible effects on connectivity in larger networks, which is one of the major areas of interest in autism research right now. Okay, so next I'm going to move on to diffusion imaging, speaking as quickly as I can. So this is just basically a recap of, you see very rapid growth in the first 2 years, 95% of maximum by 6 years, and then slower growth followed eventually by a slower rate of decline. In aging, which like I said, we probably won't get to that, but in aging, there's a point as you get out more towards 50 or 60 years of age or beyond where some of the rates of decline may accelerate, partly in pathological disorders, in say Alzheimer's or dementias, but also just as part of normal aging. White matter, in contrast to gray matter, has a fairly steady increase up into adulthood, but most gray matter structures will show a similar inverted U kind of a shape, but with different timings of the peaks. All right. So, I talked about that as anatomical MRI. Diffusion MRI is another approach. Really, it is anatomical MRI as well. We're describing the anatomy of the brain, the structure of the brain. So what does diffusion MRI really measure? All it's measuring is diffusion of water and the direction of that diffusion. It's primarily used for studying white matter. The main reason for that is just because in white matter, axons tend to be organized in parallel. You'll get large bundles of axons all running next to each other, which will tend to make water diffusion go in one direction more predominantly. And so it's just more conducive to using this diffusion imaging. Now, my picture's here. Before diffusion imaging came along, about the only way to look at white matter pathways and the large bundles that we have within the white matter, the only way to look at that in the human brain was with these very meticulous dissections. which this was really just somebody going in with a little tiny tool and peeling away the axons. But that was enough to identify some of the major bundles that were in there. We can do more precise mapping in animal models, so in non-human primates, by doing track tracing, where we actually inject a substance that's then transmitted down the length of the axon. And we can get more precise that way. But then we're also always left with the question of, well, but is this primate brain structured the same way as the human brain? And there are some differences. So how do we get from it's just the diffusion of water to the nice maps that we can get of tracts within the brain? Well, water normally is, so what we're measuring is just the Brownian motion of the water. We're looking at time scales that are only sensitive to that. But then as water molecules diffuse, they encounter boundaries. What this is trying to illustrate is here you've got several axons all running parallel to each other, and then over here you're looking at them in cross-section. And what will happen is individual molecules, the colored lines, will tend to, if they run into an axon, that will limit the diffusion in that direction. They're going to move more freely along the length of the axonal bundle than trying to cross the axons. And you actually are looking at diffusion both within and between the axons. So it's not limited to one or the other. You're looking at all of the water that's in the brain. And there's multiple different barriers that diffusing water might run into that include, I think I moved a slide somewhere else. That can include, it's going to be the axon membrane itself. That contributes to, that's one of the restrictive factors. Myelin's a very important one as it wraps around the axons. There's also effects, though, that are caused by structures within the axon. There's microtubules and neurofilaments that are part of the cytoskeleton. There's other cells that are around the area that are all contributing to the way that water can diffuse within the voxel. And so we design our MRI acquisitions to be able to sample that. And I'm not going to go into the physics of the acquisition, but just to give you an idea of what some of the standard measures are if you're not familiar. The most common method of diffusion imaging is diffusion tensor imaging, or DTI. And it describes just a basic ellipsoid model for diffusion. And estimates the directionality of diffusion. So what you'll usually describe, well, we'll describe water as isotropic, which just means equally in any direction. If it's water in CSF, it's going to diffuse freely. Or we may describe it as anisotropic, where it's got a preferred direction of diffusion. You're going to get that when you're within a bundle of axons, for instance, that are all organized in parallel. And from the diffusion tensor, we can also determine the primary direction of diffusion. That may be different from one voxel to the next, but we can figure out, well, does water tend to go this way or that way within any given voxel? And the amount of diffusion in that direction. The amount of diffusion in that primary direction we call axial diffusivity. And then diffusion that's perpendicular to that, which we would think would be kind of perpendicular to the axon bundle, we call that radial diffusivity. But the measures that you more frequently see reported are fractional anisotropy, or FA, which kind of, I always think of it as the directionality of the diffusion. Does it have a preferred direction or not? And it ranges in magnitude from 0 to 1. These other measures don't have that same kind of a limit. They're just describing how much diffusion is there. The other measure that you hear the most is mean diffusivity. So regardless of what direction, how much diffusion is there? Is it limited or is it big? Is it substantial? And you can actually have, say in CSF, you'll have isotropic diffusion because it's free. In gray matter, you've also got somewhat closer to isotropic diffusion simply because there's such complexity of the dendrite.